We're delighted to announce the first blinded independent results for our new Capture-Seq technology. As a reminder, this is a revolutionary technology that uses a completely different method for detecting cancer than normal methods, all of which simply involve sequencing the DNA that's involved in plasma. What we do is to separate out and purify the tumor-derived DNA. And what we were hoping was that by purifying tumor-derived DNA and getting rid of all of the background, we would get more sensitive detection of cancer, particularly in the early stage 1 and stage 2 patients where it really matters. And I'm delighted to say that in our first results, our best hopes have been borne out. So we tested 81 samples in total. 22 of those were from healthy persons, but the remainders were from cancer patients. These were colorectal and lung cancer patients. And we tested 18 stage 1 cancer patients, and we got a positive result for 17 of them. Similarly, we got a positive result for 26 of 27 stage 2 cancers. So we're talking about here 94% rate of detection at stage 1 and 96% detection of patients with stage 2 cancer. And for the 22 healthy samples that we tested, 21 of those were negative. So that gave us a specificity slightly above 95%. To put that in context, it's better, for example, than the fecal immunochemical testing currently used for colorectal cancer worldwide. It's patented with multiple applications protecting it, and those results are phenomenal compared to any other results that I've seen for liquid biopsy for early detection of stage 1 and 2 cancers.
I'm really excited about this breakthrough that Volition has come up with, because we are really shifting the paradigm from searching for a needle in a haystack to literally removing the haystack. This is really important from a clinical perspective for patients, because imagine that you're panning for gold. Most of the liquid biopsy companies are scanning through the entire riverbed. What Volition has managed to do is almost have a magnet that pulls out the gold from the riverbed before you even start counting it, or in our case, sequencing it. No one ever really thought that this kind of purity could ever be achieved from immunoprecipitation, and Volition has managed to do it.
From a clinical perspective, the proof of concept and early blinded validation results are extremely encouraging, and the early stage detection of 95% of stage 1 and stage 2 cancers is potentially game-changing. Detecting cancers early, before they manifest, before they grow, is a paradigm shift. The potential significance for patients and the medical community is huge. If validated in larger cohorts, CTCF Capture-Seq could contribute to multi-cancer early detection. Simply put, there is a huge unmet clinical need. We also believe that adaptation of the Capture-Seq technology will allow us to detect minimal residual disease and be extremely useful in monitoring disease progress and response to treatment. We're really excited to announce these results, really excited to be working with key industry partners, and we really hope to be able to transform and adapt this technology to bring it to clinicians as soon as possible.
What Volition are doing is they use a chromatographic process whereby they use antibodies against a well-known transcription factor called CTCF to pull out sequences of genomic DNA which is floating in the circulation, which is attached to these transcription factors. And that allows an enormous enrichment of these particular sites in the genomic DNA. And it turns out that these sequences occupied by the CTCF transcription factors are very prominent in many types of cancers. Surprisingly non-intuitive is the fact that these sequences, of course, are in every cell, not just cancer cells, such that when the DNA of any sort is released into the circulation, the factors only seem to stay bound to the particular sequences of DNA in the circulation if they have originated in a cancer cell. So this enrichment removes the signal to noise, which besets all types of genomic analysis of cell-free DNA in the blood, such that it gives a very strong signal even though there's very little of this circulating DNA. And so that's what's described in the manuscript. And what we now need to do is apply the technique to much greater numbers of blood samples.
I'm convinced that it will shake the current DNA analyses, so it really goes beyond what we are currently using today. The versatility of the platform will also allow to apply to different applications, so not only cancer diagnostics, but we can probably be able to discriminate between cancer and inflammation and all types of different pathologies. We've been testing cancer samples from stage 1 to stage 4, and when you look at the ability of the platform to discriminate between those conditions, it's just phenomenal. The team, we're currently looking at optimizing technology already to make it quicker, more affordable, and for it to be used on a day-to-day basis by a multitude of different groups.
So I'm very excited to start a round of discussion with potential commercial partner about this new technology, which has a very large range of potential application, from tumor-naive MRD to early cancer detection. And what it's worth mentioning, it's both on the human side, obviously an enormous market, but also on the animal side. It's a new class of biomarker that fits very nicely into the portfolio of both those human large liquid biopsy companies, as well as the existing portfolio partners that we have on the animal side and new ones that we're also in touch with. So we have started commercial discussion. We are getting a lot of interest, and we expect this interest will materialize into commercial agreements later this year.
Our partners we're discussing this with and potential licensees are very excited to see good early stage data, and we're really making strong progress on a number of fronts in talking to licensing partners about commercializing this platform. So a great outcome, and my congratulations to the entire team for their tireless work in getting this to actual fruition on the capture side, particularly with early stage detection. Congratulations.